Dear students, as you know, due to corona crisis and lockdown in the country, it is impossible for us to teach physically. Therefore, we have started online classes and we are teaching with the help of online videos, tutorials, assignments, etc. by using Google Class and other apps. Today we will discuss about Golgi complex. As you know that Golgi complex is also known as Golgi body. Golgi body or Golgi complex was first of all discovered by Camillo Golgi in 1898. As you know that Golgi complex is found only in eukaryotic cells, that is plant cells and animal cells. And in a cell, Golgi body never holds a fixed position. It may be near the nucleus or peripherally positioned. The functional status of the cells do govern the position of Golgi body in a cell. Example, in exocrine or secretory cells, it is lies between nucleus and secretory poles, whereas in liver cells, it lies close to nucleus. Golgi bodies the number of Golgi bodies present in a cell are greatly reflected by the type of the functions carried out by the cell. Cells that are active in synthesis generally have many Golgi bodies than those cells with less or poor synthesis. In liver cells and nerve cells, there are up to 50 Golgi bodies per cell, while in Paramoeba, there are just 2 Golgi bodies per cell. As far as size of the Golgi body is concerned, size is also not a fixed. It is related to the physiological state of the cell. They may be small in muscle cells or large in glandular cells. The Golgi bodies are enlarged during their maximum functioning and reduced in size while in low physiological state. Liver cells, salivary gland cells, endocrine gland cells provide best example of size heterogeneity at different physiological states. As far as chemical composition of the Golgi complex is concerned, Golgi complex or Golgi body consists of protein and lipids. There are about 60% protein in Golgi body and 40% lipids. This is the cis axis or cis phase of the Golgi complex, and it is the trans phase or trans axis of the Golgi complex. These are vesicles, secretory vesicles of the Golgi complex. Dalton and Flex in 1954 clearly revealed the structure of the Golgi body. Through electron microscope study. Structure of Golgi body was revealed by Dalton and Flex in 1954. Golgi body is made up of 3 major components. First, flattened sacs or cisternae. These are sac-like structures. Second, cluster of tubules and vesicles. And third, large vacuoles filled with amorphous or glandular content. Cisternae or lamellae consist of flattened parallel sac piles up one upon the other, forming stakes. The number of cisternae varies from species to species. However, 3 to 7 are most common in plants and animals. The number may go high up to 20 in some algae. Stakes of cisternae are arranged concentrically with a convex forming phase close to the nucleus and concave maturing phase enclosing secretory vesicles. Golgi complex have several types of enzymes. As far as enzymes are concerned, Golgi complex contain thiamine pyrophosphatase, acid phosphatase, peroxidase, glycosyl transferase, glucose 6 phosphatase in very low quantity, and 5 nucleosidase. As far as functions of the Golgi complex is concerned, Golgi complex have many functions. Of these, some very important functions include synthesis of polysaccharides. Polysaccharides of mucus, that is mucin, are synthesized in the sacs and carried to the surface of the cells. Polysaccharides of mucus, that is mucin, are synthesized in Golgi body. Second important function is protein transfer. As we know that protein is synthesized by the process of translation. And translation takes place on ribosomes. Ribosomes are found on rough endoplasmic reticulum. Therefore, the proteins synthesized by the rough endoplasmic reticulum pass through the cavities of rough endoplasmic reticulum into the Golgi complex and are attached to it and create zymogen granules before being discharged. Next function is secretion of glycoproteins. Next function is secretion of glycoproteins. Proteins synthesized by ribosomes enter Golgi body through rough endoplasmic reticulum. Here they combine with carbohydrate part under the action of glycosyl transferase enzyme. During the process of glycosylation. This is an important step of protein targeting that occurs here. Next function is synthesis of glycolipids. Glycolipids are combined product of carbohydrates and lipids. The Golgi body may be involved in the assembly of plasma membrane associated with glycolipids. Golgi complex also involved in immunoglobulin formation. As you know, immunoglobulins are protein play very important role in immunity. These are synthesized by plasma cells and consist of polypeptide chains and carbohydrates. And it is inside the Golgi body that these carbohydrates are added by the enzyme glycosyl transferase. Next function is absorption of lipids. Golgi bodies are involved in absorption of lipids from the circulating fluids, transformation within the cell, and transport to the cell membrane. Next function is absorption of tonic compounds. Some compounds like copper, iron, lead, and other heavy metals are absorbed by Golgi bodies. Golgi body also involved in formation of cell wall. As you know, cell wall of plants cell is consist of cellulose, and cellulose is a polysaccharide with little proteins and lipids. During cell division, Golgi body is involved in cell plate formation and finally cell wall formation. Next function is elaboration of yolk. Golgi complex in oocytes play an important role in elaboration of yolk. Golgi complex is also involved with neurosecretions. Association of neurosecretory cells with Golgi body has made it clear that Golgi bodies do help in neurosecretions. Acrosome formation in animal cells, sperm cells, it also takes place with the help of Golgi complex. The head of sperm is derived from the membrane of the vesicles of the Golgi complex. The enzymes of the acrosomes will break the egg membrane like antifertilizin and hyaluronidase are also produced by Golgi body. Lysosome formation is also takes place with the help of Golgi complex. There are enough evidences to suggest that lysosome pinches off from the maturing phase of the Golgi body. Egg cover, that is a protective cover of the egg, is also formed by the Golgi complex. In many mammalian tumor and cancer cells, Golgi complex has been described as the site of origin of pigment granules, that is melanin, and also in pigment formation in retina. Fluid regulation it also takes place with the help of Golgi complex in many protozoan. This is concerned with regulation of fluid balance. In nutshell, we can say that the main functions of Golgi complex are protein transfer, secretion of glycoproteins, immunoglobulin secretions, synthesis of polysaccharides, absorption of toxic compounds, neurosecretions, yolk elaboration, egg covering, lysosome formation, absorption of lipids, cell wall formation, fluid regulation, and synthesis of glycolipids and pigments. Dear students of MSc second sem, tomorrow we will discuss a new topic from your syllabus. I hope you are studying and stay at your home in such a situation so that you could be safe. Okay, all the best.